Great. Thank you, Rupam. Good afternoon, everybody. What I'd like to present over the next 15 minutes or so is our experience with some of the technology that Rupam had just described. And our implementation for technology like this is a little bit different than most laboratories, given that we function not only as a large sharing biorepository, but also an analytical services group that generates not only high quality biomaterials, but also data, which is distributed to a large number of individuals. So our responsibility for getting it right, so to speak, is a little bit larger than from an individual lab level. So just by a brief way of introduction, the RUCDR is currently the federal repository for 5 NIH institutes. We enable the sharing of DNA, RNA, cell lines, tissue, and data, not only for NIH institutes, but for a number of research advocacy groups and private sector clients. We have a large staff that has to be trained in these technologies, so technology implementation is a challenge for us, depending on what we're trying to do, and we have very large numbers of samples. More recently, we have built a CLIA lab within the repository for starting to expand some of our applications so that the data could be more broadly used. And the perspective that we take when it comes to every sample being treated as a limiting or precious sample is that there's a difference between just storing your samples and biobanking a sample. So for us, every sample that we take has to be considered really kind of a renewable resource, meaning that many labs have access to these samples to do their own kinds of analyses. And even if we're going to provide data on that sample, people still want those primary samples to validate and generate their own information. So the difference between a stored and a biobank sample is that a bank sample is really proactively acquired for future testing and analysis. And our partner in the area for molecular oncology is the Cancer Institute of New Jersey, and their personalized medicine initiative is really to collect every sample that comes in the door, generate a set of baseline data that can then help research as well as some of their clinical goals down the road. A bank sample is often sent to multiple different recipients. So again, we talk about having very limiting amount of samples. So the technologies that we focus on really require that minimal sample be used so we can preserve those collections. And a bank sample should ultimately never be depleted. Within our program, there are 5 major functions, with the last being analysis. But sample acquisition, the processing of that sample, whether it be FFPE, fresh frozen tissue, whole blood, what have you, then the storage and distribution of that sample is critical for everything that we apply our technologies to. So a lot of us that just look at that end stage analysis really wonder about what that sample went through to get to where we are, and then how do you effectively assess the quality of your data based on that sample. So tighter, more reproducible technologies are really critical for success. Some of the essentials that we look for, which lead into the technologies that we run, are around these functional essentials that we apply for maximizing biological resources. We have to maximally use our primary sample because there are undefined applications. If 7, 8 years ago we were talking about next generation sequencing, we wouldn't really know the quality or the amount of sample that we would need. And even in this short period of time, those requirements have dropped. And just when you thought they're about leveling off, new technologies are developed that allow us to get even further with even less amount of sample. Efficient processing, maximizing our extraction technologies, defining appropriate storage formats so we don't squander precious samples, looking at creating renewable resources, creating cell lines, iPSC lines from cryopreserved lymphocytes and other things that we could look at for analysis down the road, as well as nucleic acid amplification, and then ultimately the appropriate distribution guidelines. So when you request a sample from a biorepository or somebody requested from you, make no mistake, they're making their own little biorepository with those samples. And we only have enough sample to go around. So when we started working with RainDance, it was actually around digital PCR for an application where we were looking to dig deeper in assessing the quality of our samples. And by this I mean, when I show you our DNA workflow in a second for analysis, we were really looking at quality control based on what the available detection technologies could tell us about our sample. And that really was limited to high throughput genotyping for a large number of these samples and these very large studies. Therefore, our quality control was looking at the level of genotyping. Now that we distribute these samples for much higher resolution analyses, it's our responsibility, and it will be lots of other biobanks and those that hold those samples for all your analyses to be able to look deeper. Well, one way we were able to achieve this is by looking at digital PCR. And we looked at many different technologies that could do this, and the fact of the matter is, as Rupam had alluded to, in order for us to really say something quantitative, not just qualitative about our samples, is that we needed to get the single molecule detection. So again, this was a slide that he had shown very quickly, but I just wanted to underscore that this kind of droplet technology, when it comes to digital PCR, allows us to qualify every sample for a key set of markers before they're distributed for deeper analysis. And then more recently, we looked at the Thunderbolt's target enrichment panel, so for the cancer panel. And what this allows us to do is, again, to use very small amounts of sample and using this droplet-based technology, actually get, again, a single molecule resolution, but this time not for PCR, but at the level of next-gen sequencing. And why is this important? It's important because we can actually engineer this kind of technology into our workflow. So it becomes a standard part of the collection, extraction, analysis, and storage part of our workflow, which makes this data available, which really short-circuits the time of being able to select the samples that you now want to do whole exome, whole genome sequencing on, and really maximizes the use of each of those samples. So these are the genes that are on the panel. They're comprised of about 230 amplicons. We had looked at this panel for a variety of different sample types. And one thing to point out is that we actually looked at this, and when we evaluated it, compared to the AmpliSeq panel, the Cancer Hotspot panel version 2, which we've been using for a long time, and wanted to see comparatively what those results look like. The targets selected here, I think were very well thought out. Again, for us, it's about continuity for samples. So we have a lot of data, a lot of legacy data, and we need to know that we can compare that data for new samples on platforms like this to what exists, especially because we keep this centralized database that we're responsible for all of the users for these samples. The nice thing about this approach for this technology for us is that we actually can use the same technology to validate our next-gen sequencing target hits. So it's the same technology. The source instrument generates the droplets. In this case, the Sense for digital PCR allows us to do the counting based on our qPCR measurements. It's just as simple as setting up Thunderbolts for generating your library, but here we actually get detection based on our qPCR outputs, and I'm going to show you the same technology was actually used to validate the hits in this evaluation. When we start implementing technology, there are a lot of considerations that we take, again, because of the scale and the size of what we're responsible for doing. So we have to look at specificity. So content selection and accuracy is key. We look at the sensitivity of our technology that we're evaluating, how much starting material do we need, what levels of detection can we achieve, how applicable is it to our programmatic goals. So how flexible can we be, and how reproducible is that data? Flexibility, maximize the utility of each technology. The fact that with the Raindrop that we can validate our next-gen sequencing panels is a huge advantage. The efficiency, workflow implementation. So when you're dealing with lots of samples, it needs to be simple. How scalable is the technology? And then also cost is a driving factor for enabling global implementation. So our study design for this evaluation was pretty simple. We looked at colorectal tumor biopsies. We were very interested in different kinds of tissue. So we had a number of quad sets of samples, tumor versus normal adjacent, and we also looked at fresh frozen versus FFPE. Every day we get asked questions, say, hey, I have some fresh frozen samples, some FFPE samples. Of course, we can combine them, right? And we can compare the analysis across them. I don't think that question will ever stop being asked. Minimum sample input was used for all technologies. For Thunderbolts, we used 10 nanograms or less. For the Ion AmpliSeq, we used 20 nanograms or less. And then we were required to validate by digital PCR for some of the targets that we saw. And this is just an excerpt, a piece of data from TCGA back in 2013. So instead of looking at all of the things that we found, let's just focus on some of the mutations in KRAS and P53, which are high incidence in these colorectal cancer samples. But before I show you the data, one of the really important things to underscore are some of the workflow considerations. Again, if you're not dealing with large sample numbers now, you very well may be in the future. And this becomes a really large issue for us. So the technology is highly scalable, about 4.5 hours of hands-on time, a total of about 12 hours to get to your sequence-ready library. By comparison for AmpliSeq, there's a considerable amount more time. And this kind of adds up for the numbers of samples that you run. But one of the things that was really striking to us was the time to technology implementation. Literally in 2 days, all of the Thunderbolt's enrichment was done and on the sequencer. It took us about 3 months starting with AmpliSeq some time ago to get to that same point. And even though these technologies continue to evolve, really we find it's the simplest ideas that work best. I don't think there's anything that we could do ultimately to improve on that. So this is some of the performance data. Looking at the Thunderbolt's cancer panel results versus the AmpliSeq in those same samples. And this is really consistent with what Rupam had shown. This is obviously our data from our samples. We did see an increased number of reads per target, which was nice. And then all the way up through 100x, at least for the Thunderbolts, we saw 100%. Up to 500x, we saw very good results. We're obviously very excited with the latest revision of the panel to be able to achieve even a greater significance for performance. And our uniformity was outstanding. And Ion AmpliSeq works just fine. It's a different workflow. It's a little bit of a different approach. However, the performance, albeit what we had set was originally the gold standard now, is just not performing as well as some new technologies that exist. What you can see here is a difference at 500x, but more importantly, the uniformity. And you can see whenever you start to see differences, it's usually in FFPE. And that's the predominance of the samples that we're running for this technology or have been. And if you look at the tumor versus normal, the uniformity drops off. So if we look at some specific mutations that we were interested in, here's one of the KRAS mutations. And the other thing that is interesting is the ability to use digital PCR again on the same instrument to be able to see how tight those variant frequencies are. So again, the AmpliSeq performed very nicely. Here's the Thunderbolts, which performed also very nicely. And if we look at the variation frequency by digital PCR, it lines up. But here you can start to see some differences. So here's a sample that also had a variant frequency of about 38. This was very close. The variant frequency for AmpliSeq is not as tightly aligned. And then here is for a mutation that we found in the tissue samples from those same colorectal samples that we tested. And you can see here a tight correlation. And then there is one sample here that we detected with AmpliSeq that we didn't detect with Thunderbolts. So as we look at this, we had to ask ourselves why. And it turns out for this sample, because we required so much more DNA for the AmpliSeq panel, we actually went back to the block and had to take additional slices. And as we all know, if we can't use the same slice for analysis through and through to validation, we potentially run the risk of getting a different result depending on where we are in the tumor or in the biopsy, and that leads a lot of variability into our assessment. And this is something that we're always cautious of, but is an example of, if we can get more out of the use of each individual sample and we're able to capture everything with the first piece, that we don't run the risk of doing that in the future. And then here's some data looking at some P53 variants. So again, you can see the tight correlation between what we identified in variant frequency on the Thunderbolts and what we were able to validate on the Raindrop, and just a slightly different profile when compared to the AmpliSeq variant frequency. So we were very happy with these results, and for the first time, we were really able to compare these variant frequencies on a really more exacting level by using digital PCR as the validation in that same technology. So looking at our DNA QC analytical workflow, there are a lot of labs that do things by hand. This is more along the lines of the workflow in our lab where it's automated from DNA extraction all the way through to actually assessing sample quality using SNP genotyping. And like I had mentioned, there are certain limitations associated with the QC that you do on a sample that may be used for an analysis that has a higher resolution. And this is what a DNA workflow might look like under construction, and this is actually what it is. Where now we actually use the same chemistries for FFPE as well as whole blood and fresh tissue that are mostly bead-based. We have the same technology infrastructure. We're refining the automation associated with how we do it, but this will become an integral part of our process. So not only can we use this technology as a part of our quality control assessment to make sure that those samples are unique, that they're not contaminated, and that they will hold up for sequence-based analysis, but we can also do target enrichment and generate key pieces of data that now will be appended to those samples for distribution with them. And this is something that at the right cost, at the right workflow, can be done on every sample. So we know that there's a need defined for biobanking. That's why we have 15 to 20 million samples under management. But technology integration is critical for molecular profiling on an enterprise level. And this kind of analysis is going to be pushed at the level of the biobank and the program so that people will be able to get this data to facilitate and move on with a deeper level of research on every sample. Specificity and sensitivity is mission critical. Being able to do the analysis and validation on a single instrument is a bonus, so this integration is really important. The efficiency and cost is operationally essential, and that will be really critical in helping us do this on an enterprise level. And what we found is that the Raindrop technology enables us to integrate sample QC, targeted molecular profiling, and validation in a single platform, which again, for a lab that deals with this kind of a sample flow is really critical for success. So I just want to thank some of our collaborators from CINJ, Bob DePaula, Sridhar Ganesan, and Lorna Rodriguez, who collectively run the Cancer Institute as well as the Personalized Medicine and Translational Initiatives, Jay, Mike, and Wasi from the RUCDR. And with that, I'd be happy to answer any questions. Thank you.